To identify a microscopic threat, whether it's a banned performance-enhancing drug, an industrial pollutant, or a viral structure, laboratories rely on the ultimate chemical scale, the mass spectrometer. Inside, this machine uses precisely calibrated magnetic and electric fields to steer molecules toward a detector. It works much like a glass prism bending a beam of light, separating the chemical components by their mass so they can be identified. But there is a massive catch. Magnetic and electric fields have zero effect on neutral molecules. If you inject a raw, uncharged sample into the machine, it flies straight through the fields and hits the wall. It is invisible to the detector. To steer the molecule, we have to force an electrical charge onto it, a process called ionization. And forcing a molecule to accept or lose an electron requires transferring energy into it. Weighing a molecule is not a passive act. It is a violent physical intervention. Transfer too much energy, and you will rip the structure completely apart before it ever reaches the scale. The oldest and most aggressive method of transferring this energy is electron ionization, or EI. To use it, the chemical sample must first be heated and vaporized into a gas. Inside the chamber, the gas sample flows into a perpendicular trap, a high-energy electron beam fired from a heated filament. Operating at 70 electron volts, a striking electron transfers so much power it knocks one of the molecule's own electrons cleanly out of orbit. The sample is now a positively charged radical cation. But the 70 electron volt impact leaves it so highly energized and unstable that it violently shakes itself to pieces in a matter of microseconds. This graph shows the result of that destruction. Instead of detecting one intact molecule, the machine records a chaotic spectrum of shattered fragments. We call this hard ionization. Molecule's blueprint by piecing together its shattered puzzle pieces. EI is the perfect sledgehammer. But if you need to measure the exact original weight of the intact molecule, this method fails completely. To get that intact mass, scientists deliberately lower the amount of energy transferred to the sample. They use a softer alternative called chemical ionization. These equations show the reaction inside the chamber. Instead of blasting the fragile target directly, the electron beam energizes a massive cloud of methane gas. The methane absorbs the impact, acting as a gentle middleman that bumps the sample to smoothly hand off a proton. This bottom graph illustrates the analytical difference. By using a chemical middleman, the target sample receives its required electrical charge without the destructive kinetic energy. The structure remains whole, leaving one massive peak on the readout. Chemical ionization trades the detailed structural puzzle pieces for a definitive, unambiguous answer on the molecule's overall size and weight. Both of these methods share a hard physical limit. To analyze the sample, you first have to boil it into a vapor. For small organic chemicals, that works fine, but massive biological giants, like complex proteins or DNA strands, cannot be boiled. They simply burn and decompose. To weigh the molecules of life, late 20th century researchers like Koichi Tanaka developed desorption methods, techniques specifically designed to lift these massive, fragile structures into the vacuum of the machine without destroying them. One major breakthrough is MALDI. In this method, the fragile biological sample is mixed into a crystallized chemical liquid called a matrix, which dries on a metal plate. A focused laser pulses onto the plate. Instead of hitting the sample, the laser energy is absorbed entirely by the matrix material. The matrix acts as a sacrificial shield, superheating and exploding into a gas plume that gently carries the intact biological sample along for the ride. By destroying the matrix, MALDI effectively cheats the vaporization problem, ensuring the biological target survives the launch. While MALDI handles crystallized solids, analyzing complex biological samples suspended in water requires a different approach, electrospray ionization. Here, the liquid solution is pumped through a microscopic stainless steel capillary needle that carries a high-voltage charge. The liquid sprays out as a fine, highly charged mist. As this mist travels, a drying gas evaporates the solvent. The liquid droplets shrink, forcing their trapped electrical charges closer and closer together. Eventually, the electrical repulsion between those charges becomes stronger than the surface tension holding the liquid droplet together. This triggers a Coulombic explosion. The droplet violently bursts apart, leaving behind perfectly intact, naked protein ions floating in the vacuum. Electrospray ionization allows scientists to extract incredibly fragile biological structures directly from a wet, natural state, ready to be weighed by the magnetic fields. A mass spectrometer is the most precise chemical scale we have, but it is entirely dependent on the specific ion source bolted to the front of it. Chemists can intentionally use the brute force of electron beams to shatter unknown synthetic chemicals to map their structures, or they can rely on the delicate choreography of electrospray to float intact viruses and proteins out of liquid droplets. Our ability to map the invisible molecular world ultimately comes down to one thing: mastering exactly how much energy we use to touch it.